Good afternoon, everybody. Thank you for joining us today. My name is Chris Grange, and I'm from Cell Signaling Technology. And together with Dr. David Rimm, we're going to be talking about spatial imaging as a tool for discovery of new candidates to better predict response to immunotherapies. The first question to ask is why would you want to understand spatial biology, right? So you can discover new cell types, understand the functions of those cell types, learn how the cells are impacted by their neighbors, and how the cells organize and interact to influence disease progression. And you might say, well, you can answer some of those questions possibly with single-plex chromogenic IHC. But with that assay, what you end up with is many slides, many tissue sections, each stained with a single antibody. And it's hard, if not impossible, to take all of that information on multiple slides and put it together in a meaningful way to answer some of these more complex questions. What spatial imaging does is it takes those multiple slides, each stained with a single antibody, and it replaces them with one tissue section stained with multiple, sometimes dozens of different antibodies. And this allows us to answer those complex questions about disease progression and treatment. So what's required to understand spatial biology and perform spatial imaging? Well, the first thing is you need advanced platforms and some pretty sophisticated image analysis software. And Dr. Rimm is going to talk about that after I get done. Today, I'm focused more on what I think is one of the most important building blocks of a spatial imaging assay, which is high-quality IHC-validated antibodies. And for all of these spatial imaging platforms, or I should say all of these spatial imaging platforms require directly conjugated primary antibodies. And in order to conjugate an antibody, you need a special carrier-free formulation of that antibody. And so I'll just note that at CST, we have the ability to offer nearly 100% of our monoclonal antibodies in our catalog in carrier-free formulations. And I will also note that antibodies that are well validated in FFPE tissue using chromogenic IHC are very, very likely to work in any spatial imaging panel and on any of the various platforms. So for the rest of this talk, I want to get into how we validate antibodies at CST and talk about our philosophy. So just like there are the hallmarks of cancer, we at CST have come up with the hallmarks of antibody validation. And these are strategies that outline our philosophy for optimizing and validating antibodies. And today I'll be talking about IHC, but these strategies can and should be used in any application that uses antibodies. So the first strategy is a binary strategy. This is when you have a model with a confirmed expression of your target of interest and another model where you know that your target of interest is not expressed. Here we're looking at ADORA 2A, which is a target that's expressed exclusively in the striatum of the brain. So this is a pretty easy target to validate, right? Because expression is so limited. When we look at the binary model, we see nice staining in the striatum of the mouse brain, and the rest of the mouse tissues that we've tested are clean. But as you all likely know, there are many more difficult targets to validate, and for those targets, we require more complex strategies. Another binary strategy I want to talk about is knockout models. So here you can see Fc gamma receptor beta antibody in 3 different applications stained on wild type versus knockout mouse. The point I want to make about knockout validation data, though, is that it's just a single data point. In this example, we can see 2 clones directed against 53BP1, both of which show an appropriate band in the wild type cells and no band in the knockout cells. So both of these antibodies pass in this model. Furthermore, we can take those same cells and put them into FFPE cell pellets, and again, we see that both clones pass. So if you are using just this single data point, you might think both of these clones can be used for IHC in FFPE tissues. But when we look at the tissues, you can clearly see the clone on the left has nice, strong nuclear staining as expected, whereas the clone on the right shows weaker nuclear staining with cytoplasmic and membrane non-specificity as well. So if you were only using that knockout data, you might think you could use these clones interchangeably in tissue. So the point is, knockout data is not sufficient in validating antibodies, especially in IHC. So moving on to the next strategy, we'll talk about ranged expression. It's not always possible to have a binary model, or it's not always cost-effective to create a knockout mouse, say, just to validate an antibody. So in cases like that, we can use ranged expression, where we have known target levels over a range of different tissues or cell pellets, and we can compare that data to what we see in IHC. As you can see here with CD39, spleen has high RNA, and we see abundant staining. Heart has much less, and we see less, and skeletal muscle is nearly negative. Similarly, we could use an orthogonal strategy, and this is using non-antibody-based assays as a comparison to what we see in IHC. So at CST, we're able to leverage our expertise in mass spec to categorize some of our frozen and FFPE tissues and compare that data and use it to validate what we see in IHC. Here you can see a mass spec study looking at small cell lung cancer, and we've chosen a low, medium, and high expressing tissue, and that compares favorably with the DLL3 IHC staining. Next, we have a multiple antibody strategy, and this is one of our favorite ways to validate antibodies at CST. So you can see we have here 2 antibodies directed against different portions of the Nectin-2 protein. And so when we stain those antibodies on tissue, if we see overlapping signal, we can be fairly confident that that signal is specific for Nectin-2, because it would be exceedingly rare for 2 antibodies directed against different epitopes to show nonspecific staining in the exact same cell types or areas of the tissue. And I'll note here that the second antibody you use, it doesn't have to be perfect. It can be a poorly performing antibody, but as long as it detects the target of interest, even if there's nonspecific staining, you can always compare the staining where they overlap to help validate your more impressive IHC antibody. Next is a recombinant strategy, and this is basically using overexpressed transfected cell pellets to validate antibodies. And on its own, a transfected cell model is kind of the lowest bar for antibody validation. Even if you have a really poor antibody, you're likely to see signal just because in a transfection there's so much protein present. But it can be used to determine if antibodies will cross-react with closely related family members. Here we see a HER3 antibody stained on EGFR, HER2, and HER4 transfected pellets, and clearly we've shown that this antibody does not cross-react with these family members. Finally, we have a complementary strategy, and this would be used for antibodies directed against post-translational modifications. This would be things like lambda phosphatase treatment and peptide competition assays. So here you can see with the phosphoglycogen synthase antibody, staining is completely absent in the lambda phosphatase treated tissue. So this proves that the antibody is phosphospecific, but it does not prove that it's specific for phosphoglycogen synthase. All of that staining that you see, it could be nonspecific staining on different phosphoepitopes of different proteins. Similarly, with the trimethylhistone and non-phosphobeta-catenin antibodies, we see that antibody staining or binding has been completely blocked in the presence of the trimethylpeptide or the non-phosphopeptide, and that the non-methylpeptide and the phosphopeptide have no effect on antibody binding. So we've again proven that the antibody is methyl or non-phosphospecific, but further validation is required to prove that it's specific for our target of interest. So those are the 6 hallmarks of validation, and I just want to talk briefly about another sort of hallmark of what we do at CST, and that's species reactivity testing. So as you're likely aware, if you have an antibody that works well in human tissues, there's no guarantee that it's going to work in other species, such as monkey or mouse. And at CST, when we have an antibody that we believe is human and mouse reactive, we'll do that testing up front. So if it shows reactivity by Western in human cells and in mouse cells, we'll test it by IHC. And we'll put that information on our website so that you don't have to worry about it. But we've recently started to do a different type of species reactivity testing because we became aware that many of you and many of our customers are using patient-derived xenografts and humanized mouse models, right? And so in this case, more specialized validation is required. So say you wanted to look at CD4 in a patient-derived xenograft. We have this great CD4 clone. As you can see, reactivity is listed as human, but that doesn't necessarily mean it doesn't stain mouse tissues. There's a chance that it could react weakly with mouse CD4, and there's a chance that you could see non-specific staining in mouse tissues that's not present in human tissues. So what we've been doing is we've been going back and looking at some of the most common immunotherapy targets and testing them in species for which they're not predicted to react. So in this case, we tested it on a number of different mouse tissues, and as you can see, it's completely clean. So this would be a good candidate for patient-derived xenograft studies. And I just wanted to leave you with this statement, which we always say, that no single assay is sufficient to verify the specificity of staining, and also to reiterate that all of our antibodies that are validated and recommended for IHC are very likely to work on the spatial imaging platforms that you'll be hearing about this week. And so with that, it's my pleasure to introduce our longtime collaborator, Dr. David Rimm. Dr. Rimm is a professor in the Department of Pathology at the Yale University School of Medicine and is board certified in anatomic pathology and cytopathology. His lab group focuses on quantitative pathology and spatial imaging, with projects related to predicting response to therapy, as well as recurrence and metastasis in several types of cancer, including breast and lung cancer, melanoma, and head and neck squamous cell carcinoma. We're excited to hear about some of Dr. Rimm's latest work in digital spatial profiling and his perspectives on the importance of antibody validation in both high and mid-plex tissue-based assays.
Okay. Hi, thanks, Chris. Thanks for warming everybody up here and talking about antibody validation. Certainly that's near and dear to our heart. And I'm not going to talk about it today since Chris did a great job of covering the key things, and we actually use a lot of those. We don't call them hallmarks, we call them pillars of validation in our lab, and we worry about that a lot, unless, of course, we're using cell signaling antibodies, and it's already done for us beforehand. But what I'm going to talk about today is spatial imaging as a tool for discovery of new candidates to better predict response to immunotherapy. Even though this is not CME, these are my disclosures. And this is the outline for the next half hour or 45 minutes. I'm going to start with talking about measuring protein on slides, because most people think about measuring protein on slides as cell segmentation and then measuring protein per cell. And that's by far the most common way of measuring protein on slides. That's not what we do. Actually, we do that, and we use the various technologies for that, mostly Inform. But we're fond of measuring the protein or tissue on slides, which requires a different kind of separation or molecular compartmentalization of tissue, and I'll talk about that. And then we'll move to talk about one of the things that cell signaling wanted me to talk about was what are the chances that we'll see some of these high-plex or multiplex fluorescence in the clinic. So this is a new piece of a talk that I made just specifically for this session, talking about what Scott Rodig has done a little bit in Boston, the first place that immunofluorescence has entered a CLIA lab. And then I'll tell you a little bit about our progress, although we're not there yet. Then I'll talk a little bit about high-plex discovery tools and a sneak peek about a couple of biomarkers that we're pursuing for immunotherapy. So laboratory medicine in clinical pathology, there's lots of quantitative assays. But all the immunohistochemistry assays in anatomic pathology are binary. They're either positive or negative. That's the chromogenic assays. And so some assays are semi-quantitative, and you could even argue that there are assays that are quantitative because they count the number of cells that are positive or negative, but actually they're binary. They're positive or negative. So that's not truly, the bottom line is there are really no continuous quantitative assays in anatomic pathology. And I wanted to sort of show why that is, and then talk about the potential for quantitative fluorescence to bring those assays in. So this is an assay, sort of a standard assay curve, and you got a limit of quantification, a limit of detection, and then you want your assay to be in this range so it can be truly quantitative. But what happens in pathology is that vendors who sell assays into the clinical world make them like this, and that is that they're either negative or they're positive, and there's very little, if any, linear range. And that's because most of what we do with the 600 or 800 or 2,000 antibodies we use in anatomic pathology are binary. They're used as a diagnostic tool. If they're positive, then they're positive, and if not, you want them to be negative. And so you make a curve that looks like this. But when you measure tissue, there's different approaches to it. And this is what I wanted to talk about just briefly, that if you're measuring tissue, you don't necessarily think about measuring cells. The concept of measuring cells is commonly called segmentation, and it divides the cells, and you can even figure out how you're going to divide each of these cells into a specific cellular compartment. Some people use a nucleus and then dilate out from the nucleus. Other technologies are using other sorts of phenotyping, but start with a definition or a segmentation of what's a cell. The problem with that is that then you don't know what's not a cell, and a lot of the things that are not a cell. And so the alternative way to do that is the way that we've been doing it for years, is to measure tissue, which is another term for that is molecular compartmentalization instead of segmentation. And there we define regions by molecular interactions. And so you do the sum of the target intensity of whatever you're trying to measure divided by the pixel area. And so it's more like a biochemistry of the cell or of a tissue, where we return a value of attomoles per square millimeter as opposed to cells positive, which is more familiar to pathologists, but binary and allows you to have less continuous information. So this is an example of a fluorescent image, and this is an example, a nanostring from a nanostring molecular compartmentalization, even though they call it segmentation. It's really molecular compartmentalization. You can see the compartments here. Cytokeratin is one compartment, and that's green. CD45 is the lymphocyte compartment. You can see some here. And then CD68 is the compartment that is covered in yellow here, and those are the macrophages. Or you can combine keratin and everything not keratin and call that stroma. And we frequently do that in the lab to define tissue, because stroma is really hard to segment. In fact, I have yet to see a program, any software, that can segment fibroblasts. For all you segmenters in the audience, I challenge you to segment fibroblasts consistently. So here's an example of bright field segmentation, and we often use this software. This is QPath software, and this is an example of segmentation followed by phenotyping, so that you can identify each type of cell, and then you can count the cells as positive or negative. And we do that, and there are times when segmentation and cell counting is the best tool for the job, because you want to know how many CD8s there are, how many CD4s there are, and you don't really care how many attomoles per square millimeter there are. So I'm not saying that one is right and one is wrong, but rather that there are two different ways to think about quantification, and you should become fluid in both, in my opinion. This is probably the most progressive segmentation that has yet been done, and that's this astropath technology that was invented by Alex Saleh, who's an astrophysicist. And this came out in the science paper led by Sneha Berry last year. It's all from Janice Taub's lab. And this uses multiple passes of the fluorescence, or multiple images, in order to segment. And you can see a classic segmentation error here. And then when you do multiplex, you get much better segmentation. This is wonderful, and I think that it has a great future, but unfortunately, unless you work at Johns Hopkins, it's not available to you yet. Keep your eye on that space. This might be ultimately the best segmentation solution. Another problem with segmentation is it's highly dependent on antibody concentration. And Chris talked to you about how important it is to validate antibodies. I would argue it's equally important to find the optimal signal to noise, because you can see here at different antibody concentrations, you get different numbers of CD4 cells. So if you're segmenting, the CD8 is constant, the CD4 antibody we've diluted out. And so the CD4s that you see down here are a lot fewer than the CD4s that you see over here. The red is the CD4, the yellow is the CD8, where you can see the yellow is constant, the same antibody concentration, the changing the antibody concentration. So if you want to have a lot of CD4s, use a high antibody concentration. If you want to have a little, use a low antibody concentration. But the problem is which one's right. And that I would argue you don't know unless you've optimized the signal to noise for your CD4 in this case, and then have some sort of standardization method. I'm not going to talk about standardization today. I'm a big proponent of standardization. We'll talk about that another time. But I think that this is one of the huge pitfalls of segmentation, is that you don't know if you're right or not. And various different people in different labs can get different results by having different concentrations of the antibody, or even thinking they're using the same concentration of the antibody, but one is 6 months old, that's been sitting in the refrigerator for all that time, and it's lost some of its reactivity. And so this is a big a word of warning for segmenters who count lymphocytes. This is just a follow-up on that, showing the difference of the CD4 count, as you can see, was the same, but the CD8 count changes by an order of magnitude, even though the concentration of the antibody only changes twofold. Be careful of that. So just to compare the two, segmentation is measures cells. It's a cell-centric or cellist view of the world, that everything happens in cells. And molecular compartmentalization, which measures tissues or attomoles per square millimeter, can't count. So that's the biggest disadvantage. It can't count the cells on one hand. On the other hand, it measures tissue. And I would argue that what's interesting generally is tissue, because function comes from tissue, unless you're talking about a defined set of cells in cell culture or in blood or something like that, where the cells are really the driving force. In tissue, the stroma and the matrix between the cells and fibroblasts and smooth muscle actin and lots of things that go on outside of the cells can affect the biology. And that's why I think it's important to measure tissue sometimes, not just cells. And the other thing that worries me about measuring cells is this failure to segment or failure to optimize your titration. But not to say that one is necessarily better than the other, but I would argue you need to use both. The problem with molecular compartmentalization is it hasn't been very generally available. The Aqua software, which we invented in my lab 20 years ago, is not commercially available. However, it can be mimicked with QPath. QPath is an open source software which can measure tissue as well as measure cells. And this is the case for measuring tissue, that this is, what tissue looks like isn't a jar of marbles. This is what tissue looks like, with different size cells and little blebs coming off the cells. And so if you imagine a cross-section through here, you could see that, first of all, you might take a grazing view through the Arctic Circle of this somewhat round cell, and you would never see the nucleus, so you wouldn't call it a cell if you hadn't seen the nucleus. Or you call it a cell by some phenotyping method, but you'd still have an insufficient measurement of the area, and other parts would be missed completely. And so this is one of the concerns I have about technologies that strictly measure cells. So can we take these things to the clinic? Well, this is what we do in the clinic now, One-Plex, and this is actually, we're supposed to count, and when we measure PD-L1, we're supposed to count the number of cells that have PD-L1 staining, and you can see that that would be very hard, and particularly hard here, where these are macrophages that are staining, that have intercalated between the cells, and you can't see the border of one cell from the next. So how can we count these as we're instructed to do to ultimately predict response to therapy? And so maybe the best way to do that is to move beyond the clinic and use IF and use quantitative immunofluorescence, either with cell counts or and phenotyping, or with tissue measurement. And already IF is in the clinic in renal path and derm path, but we don't really use it for anything more than a binary tool there. Quantitative IF was introduced in Genoptix Spec about 7 or 8, 10 years ago, but it didn't stick for business reasons. But there is a group now that's led by Scott Roda at the Brigham, and they call their profile Immunoprofile, and they're using the Polaris platform and a Akoya antibody kit to actually bring immunofluorescence into the clinic, and they've done the first 2,000 specimens. He's preparing the manuscript on that, and I've seen it presented once, but it hasn't been broadly presented yet. But I think that may be what they're doing is the killer app. That is that immunofluorescent, or the response to immunotherapy probably is not a binary question. As you could see with PD-L1, we really haven't gotten the answer. And so a non-binary question, like the microenvironment of a tissue that has been treated with immunotherapy, is potentially a killer app that could bring IF into the clinic. And to quote Scott, he said, when the assay is working in your research lab, you're 5% of the way there, which is probably very true. It's very hard to bring a research app into the clinic. So this is from the immunoprofile that Scott does, and this slide is courtesy of Scott, and you can see some of the markers that he's done. The highlights are that it's started as a research cohort, which I think is the way to do it, a big prospective research cohort. You can see the panel here, and the clinical validation of the panel is extensive in his talk, but I'm not going to give that whole talk again here. It's in a CLIA-compliant atmosphere at Brigham and Women's, and the results are reported out in a mechanism that the clinicians can actually see the results, although it's not technically in the patient's chart. The results are not in the patient's electronic medical record, but it's a click-through. So you go to their medical record, and it's a link from their medical records. So clinicians can see it, although it's a research use only test. This is kind of the pathway and the overall flow from Scott's lab. And you can see that it's a pretty complicated set of events that has to occur, including automated staining, image acquisition, and download. And their turnaround time is between 1 and 3 weeks, and they have quite a number of staff in order to achieve this on the Inform platform. This is a quick glimpse of where we're going. We're trying to do a quantitative HER2, a low HER2 range. And this is actually work from Mirto, who's in the audience, to find the limit of detection and the limit of linearity, limit of detection down here and limit of linearity for HER2 in the low range. And we hope to bring this assay into our clinical lab via this pathway, where we get the slides, a pathologist circles the region to be scored, and then we go through a pathway using standardization, which I'm not going to go through right now, but then ultimately can return a report in which the regions that are circled by the pathologist have an attomole per square millimeter value, and then the pathologist can review that report and sign out the case. This is an example of one of those cases. So here's the pathologist part, and here's my little circle up here, and everything under the circle was actually quantified and shown by a heat map here. And then on the next slide, you can see the concentrations in attomoles per square millimeter of each of those fields of view. And so I can summarize this, and I might sign this out as a low HER2 case at 8.16 attomoles per square millimeter on average through 17 fields of view. This is something that we hope to bring online possibly as soon as July 1. And I would add, actually, because of where I'm speaking today, we're using the cell signaling antibody 2098. So now let's talk about High-plex. Some of you might have come here to talk about High-plex tools, since that was what was in the title. And there are lots of different High-plex tools out there, so many that I've started to lose track. And I'm specifically only going to talk about High-plex protein, because there's been a lot of activity recently in the High-plex genomics, RNA and DNA space. And those High-plex technologies tend to have insufficient resolution to ask the kinds of questions we want to ask with proteins. And so for that reason, I've looked at methods that either do molecular compartmentalization or methods that can get me down below a micron resolution. Most of the technologies are at 10 or 20 or 25 or even 50 micron resolution, which is great if you want to look at different parts of the brain, but not so good for cancer biology. So the cycling, there's the High-plex, I define as at least greater than 10-plex, which is kind of mid-plex, but mostly between sort of 10 and 50, and then High-plex is 75 or greater. And so most of these techniques depend on either cycling, which is still a pretty common technique, mass spec, MIBI and IMC, or the DNA tag-based High-plex. And this is the one there seems to be more and more of these sprouting up, and we'll have to closely watch that space. This is one of the first cycling technology actually was developed by GE, which is a kind of a interesting technology, because every time you cycled, you got slightly less antigen because the antigen suffered from the removal of the antibody. And so the cycling technology worked, but they couldn't get beyond 9-plex by cycling. But nonetheless, I want to give them credit reluctantly, because I actually presented cycling to them and told them it didn't work, and then they did it and patented it and sequenced it and published it. But this technology has actually now gone to neogenomics and still I believe the highest they've gone with this is in the range of 25 or 30. This is a really cool one that Sonia Noddick invented at Mount Sinai. And the idea here is that you go through and you do antigen retrieval and block and stain, and then you look at the chromogen, you take a picture of it, and then you wash the chromogen out, essentially. By chemical destaining, you can actually get rid of the chromogen, and then you can do it again and again and again. It's very time-consuming, but you're staining the exact same cells multiple times. And it also diminishes the antigenicity over time, so it can't be quantitative, but it can answer a lot of interesting questions. So you could see the fact that by 7 destainings, you're starting to see loss of some antigenicity, but the localization and counts can be pretty accurate. And so an interesting method that uses a chromogen. Peter Sorger is known for his CyCIF method, which is also a cycling method, but does 4 channels at a time. So you can do a number of cycles and times 4, and then you can map back to the positions, and you can high-plex pretty well with this method. And I think it's a really fine method of high-plexing, but it's complicated and time-consuming, so I don't see it necessarily getting to the clinic. And then there's Codex, and Codex is a method that gets rid of the problem of exposing the antigens to multiple conditions, because you put all your antibodies down at once, and then each has a different DNA tag, and you cycle only the fluorophores instead of having to cycle and damage potentially your antigens. So I thought this method was really cool, but the problem is it works really well on frozen sections, but not so well on FFPE, because there's no amplification step. Now, this has been since commercialized, and you can go see the Akoya booth out there, and they claim they've gotten up to 100 multiplex with this method. And then there's a couple that I don't know much about but have come along, and so I'll just mention their names here, the Miltenyi Biotech Maxima and the Lunaphore Comet. In fact, this meeting is the official introduction of the Comet, as I understand from their marketing material. I haven't gotten a chance to talk to them yet. And these are, again, cycling methods that I think have potential in specific mechanisms, but probably not likely to make it to the clinic. Then there's the mass spec IHC, and this is something that I think is really fascinating because it's a high-plex without having to use a fluorophore. And the advantages of not using a fluorophore is that you don't have the same kind of overlap you get in a fluorophore. And these are the inventors and founders of the Ion Path company. Mike Angelo here and Harris, I'm blocking on his name. And this was the beta unit that they use, although I'm sure there's a lot more going on now with MIBI. And the advantage of this technology is you don't suffer. You can have multiplex up to 50 channels or so, although I think most of them are in the 30 to 50 range, without having the spectral overlap that you see when you have fluorescence. They also have some of the techniques don't have great resolution, but this has submicron resolution. In fact, 10 angstrom resolution, a tenth of a nanometer. So that if you want to be high resolution, this is the way to go. This is just some from their paper. You can see that it's been around for a while, but they were able to ultimately make this work and commercialize this technology. It followed on the heels of this technology, which preceded that technology, which is imaging mass cytometry, that used laser ablation instead of ion beam ablation. And so lasers are harder to focus. And so the disadvantage of this compared to MIBI is that it's got lower resolution, but the resolution is still pretty high at 1 micron. And so this is still a good method to high-plex, and it still takes advantage of that metal conjugated antibody approach that you can allow you to high-plex up to 35 or 50, and then map back to where the antibody and the metal was blown off of the surface of the slide. And they actually came up with this histocat software to allow the mapping back, and a number of labs, including Kurt Schalper, our neighbor, and our lab itself has used this technology for a high-plex. The last one, and the one I'm going to talk about, and the one my favorite is digital spatial profiling. And this one works by taking an antibody and then linking it with a photocleavable linker to a unique single-stranded DNA tag, and then recognizing the tag attached to a barcode to count how many you have, but it doesn't create an image. So while this technology I think is pretty cool, and here's how it works, is that you have that barcode on the antibody, and then you define a region of interest, and I'll talk about that in a second, but then you shine the laser just on that region of interest, the barcode comes off, you sip it up in a little sipper, and then distribute it on a 96-well plate, and then you know where it was sipped up from, but you only know that region of interest. You can't generate an image. And so as a result, you can do a phenotypic, or what I would like to call molecular compartmentalization, to define your regions of interest. And I think this is a great tool for discovery, because you can tell the region of interest even though you can't tell which cell it is, and then you switch back once you've found interesting candidate markers, and switch back to quantitative fluorescence, where you might only do 3 or 4-plex, but you then have the sub-micron resolution that we've come accustomed to with quantitative fluorescence. So this is kind of a summary of the technologies, and I think it's really important to think about the resolution. And light microscopy or fluorescence microscopy can exceed 0.2 micron resolution, but in general, we're using 0.2 micron resolution. The exception to this is MIBI, which is down to 0.01 microns, and then the exception in the opposite way is DSP, which doesn't have great resolution. The laser spot's about 10 microns, and so your regions of interest are never really resolution limited so much as it's not a segmentation event, it's a molecular compartmentalization event. And so when will High-plex be in a clinical lab? I told you that there's that quantitative fluorescence is already in Scott Rodig's lab and soon will be in our lab, and those are CLIA labs. But in terms of High-plex in CLIA labs, I don't think it's going to happen. If you think about what's going on now in the research world, there's more new High-plex methods every year, and I would be remiss to not mention 10x because they have lots of High-plex methods for sequence, and rumor has it soon protein High-plex methods. And they're more and more quantitative, they're getting faster, and they require more computational power. But interestingly, while all this progress is going on in the research world, in the clinical world, we're going backwards. We're seeing FDA approval of assays that are single antibody assays, IHCs with single antibodies, and they're not reproducible. So instead of bringing this to this, we're kind of going in opposite directions, which is why I'm concerned that we're going to have to. It's not a scientific barrier; it's a political barrier to getting more quantitative work into the CLIA lab. And I think what Scott's doing and what we hope to do in our CLIA lab with just 3-plex or 4-plex, or in Scott's case 6-plex, is probably the best we'll see in the near future. And maybe someday we'll take some of these high-plex 30 or 50-plex to the CLIA lab, but I don't think it'll happen anytime soon. So the last thing I wanted to talk about, and then we can have a little discussion or ask questions, is just show you some of the biomarkers for immunotherapy that we've discovered by High-plex. And I'm not going to spend a lot of time on this. This is Mirto's work looking at CD66 and the CD45 compartment. So again, this is done on a nanostring. It was done with a 78-plex on the nanostring, and you can see that then we took it and we discovered on the DSP, and then went back to quantitative fluorescence, and we could see that this marker is associated with resistance to immunotherapy in our training cohort. Then we used a validation cohort. These are different cohorts. This one's more recent. This is older cases, all treated with immunotherapy. And you can see we saw the same pattern. And then in an untreated cohort that precedes both of these cohorts, but just lung cancers that were not treated at all, you can see that CD66 is not a negative prognostic marker, which is why we think it's associated with resistance or poor outcome in immunotherapy. In a similar work, Sandra Martinez-Murillo in the lab found a similar pattern with both a training set, a validation set, and then a historical cohort, suggesting that CD95 might be associated with resistance to immunotherapy. And so, as you can see, once we have our fluorescence CLIA lab up and running, we hope to be able to predict resistance to immunotherapy in assays 3, 4, and 5 that come out of our CLIA lab in the future. It's harder to look for biomarkers of response when the marker's lost, and so these are all markers that are lost. And so beta-2 microglobin is a classic. If it's mutated, immunotherapies don't work. And same thing, if there's loss of beta-2 microglobulin, immunotherapies don't work. But measuring loss is much harder than measuring gain. And so I doubt that we'll see, I'm less likely to see those assays ultimately come to the clinic for response to immunotherapy. And so the summary, the measurement of protein biomarkers beyond IHC is not in the clinic. It seems to be moving in that direction, but probably won't do so with High-plex. I think High-plex is still a discovery method. There's lots of High-plex methods. Most are still under 100-plex for protein, although there are thousands of plex or higher for nucleic acids. And the new ones work with DNA or RNA, and I didn't mention those. Since it's hard to even bring a 3-plex to 6-plex in the clinic, High-plex seems unlikely to get there, but they have great potential for biomarker discovery. So with that, I just show a picture of the group that did some of the work I showed and disclose my support. Then we're required to show this slide as well at the end of our Spotlight theater. Shows that we are presenting in a Spotlight theater as opposed to a general session. And so with that, I think I'll stop and take questions. Thank you.